We have all heard and have even experienced many diseases and disorders that are said to be a result of genetics. We have probably all wondered how and why this occurs. In today's lesson, we will discuss the properties of genetics and the effects that it has on human disorders. This lesson has been prepared and aimed for AP students. The objectives for this lesson are to explain and understand what genetics is, to review background information of genetics, to distinguish the characteristics of human genetics, to explain the different areas of genetics, and to explain the different diseases affected by human genetics. We will also discuss and explain how genetics can play a role in drug development. In order to begin this lesson, we're going to start with the basic question of what is genetics? Genetics is the study of heredity and focuses on genes. Genes are the basic units that are responsible for heredity. Genetics can be recognized as a study of genes and its relation to cells or the ways that they are transmitted from parent to offspring. In order to better understand this concept, let's go a little further into the background of genes and genetics. The picture we are viewing on this slide is the DNA molecule. Genetic information can be found on DNA molecules. This information is encoded on 4 nucleotides: adenine, thymine, guanine, and cytosine, and they are represented by the letters A, T, G, and C in the picture. Each DNA molecule is a double helix that are composed of a pair of anti-parallel complementary DNA strands. These DNA strands are held together by hydrogen bonds between base pairs. The double helical model of the DNA molecule was created in 1953 by American biologist James Watson and English physicist Francis Crick, names that we should all be familiar with. The 2 complementary strands of DNA allow each strand to act as a template for the other. The production of 2 identical molecules by DNA replication allows genetic information to be copied and passed on from a cell to a daughter cell. This also allows information to be passed from parent to offspring. Let us dive further into the topic of human genetics. Human genetics focuses on the human genome. This study focuses on areas such as gene mapping, gene function, and inheritance. Human genetics also focuses on detecting mutated or non-functioning genes. The Human Genome Project, which was initiated in 1986, led to rapid advances in acquisition of genetic information. Let us turn our focus to other areas of genetics that can be credited by the Human Genome Project. There are several areas of genetics that we can credit the Human Genome Project for creating. There are bioinformatics, proteomics, toxicogenomics, medical genetics, gene testing, gene therapy, and pharmacogenomics. Let's start with bioinformatics. Bioinformatics is an area that helps organize, store, and analyze genetic biological information in a precise manner. Proteomics is the study of genes and their functions. This involves the study of protein expression. Toxicogenomics is an area concerned about human genes and how it responds to toxins. Medical genetics recognizes defective genes, that are non-functional or malfunctioning and causes diseases or tissue malformation. Gene testing takes a sample of blood or bodily fluids and examines it to detect a genetic anomaly which can lead to genetically based disorder or disease. The Genetic Information Non-Discrimination Act of 2008, or GINA, was passed by Congress that restricted discriminating against an individual based on their genetic information. Congress hoped that this would encourage individuals to seek genetic testing in an effort to increase health outcomes. Gene therapy uses recombinant DNA technology to cure diseases or disorders that have a genetic basis. And pharmacogenomics is the branch of human medical genetics that deals with the correlation of specific drugs to fit specific diseases in individuals. Now that we have covered the basics, let's turn our attention to human diseases and disorders that are affected by human genetics. Here I have listed some neurodegenerative diseases, Alzheimer's, ALS, prion diseases, and Parkinson's diseases. But we all recognize that these are not the only diseases that are considered to be affected by human genetics. There are several neurodegenerative diseases that are affected by human genetics. Most neurodegenerative diseases are recognized by onset in adult life. They also involve characteristics such as distinct clinical phenotypes and cellular abnormalities. A lot of times when individuals are experiencing these diseases or the onset of these diseases, you usually start recognizing changes in their behavior, their attitude, even simple things like their posture. These things help us to recognize when our loved ones are experiencing one of these diseases. Some of these diseases can be fatal. ALS, which is one of the most common adult onset motor neuron diseases. Muscle weakness and atrophy are the common characteristics of this disease. 10% of ALS cases are familial, and inheritance exhibits an autosomal dominant pattern. Alzheimer's disease is the most common cause of dementia in adult life. Most of us have experience with someone in our family or neighborhood that has experienced Alzheimer's disease at one point or another. It is often associated with the selective damage of brain regions and neural circuits critical for cognition and memory. One of the first things you notice, or I tend to notice about someone who is dealing with Alzheimer's, is their confusion or their memory loss. That's one of the first signs that you recognize in someone that's experiencing Alzheimer's disease. Parkinson's disease is a brain disorder. This disorder is characterized by shaking, stiffness, and difficulty with walking, balance, and coordination. Autophagy lysosomal pathway, according to human genetic data, are central in Parkinson's disease. These diseases are very serious and are commonly linked to human genetics. Genetics and neuroscience can sometimes be complicated because it involves detecting weak signals. There are areas of research, such as the number of phenotypes or the number of genotypes collected, that make this research difficult. But if this is done, a direct correlation between the two would be easier to find and it would be a tremendous help in understanding how genetic affects neurodegenerative diseases. We are now going to shift our focus onto another disease that is associated with human genetics. While doing my research, I found this topic to be interesting because most people don't associate this with human genetics. Now, fatty liver disease is characterized by hepatic fat accumulation. This is mainly due to metabolic derangement and excessive alcohol consumption. Abdominal overweight, obesity, insulin resistance, and type 2 diabetes are all risk factors for the development of fatty liver disease. This metabolism is also a factor associated with fatty liver disease. Studies suggest that the key mechanism of this metabolism is not a fat accumulation, but excess liver content. Genetic factors might be directly associated in controlling pancreatic beta cell function. This can lead to helping in the treatment of this horrible disease. While doing my research, I also thought about the question of how can human genetics influence drugs and drug development. A lot of times we hear about diseases such as Alzheimer's or Parkinson's or even like sickle cell anemia, which is also a human genetic disorder. It's often so that there is not a cure for the diseases, but medication has been developed to help treat the diseases and disorders. So I dug in this and research to figure out what role can human genetics and research do into drugs. Genetic differences between individuals affect their susceptibility to disease, resistance to infection, and response to drug treatment. So drugs that may work for me or my family members may not necessarily work for others based on genetics. I did research on something called drug repurposing. And drug repurposing involves identifying single or combinations of existing drugs based on human genetics data and network biology approaches. This approach can potentially increase the speed of drug discovery at a lower rate, meaning we'll be able to produce drugs faster, but the cost won't be as high. Drug repurposing has genetics, network biology, and network polypharmacology all working together in order to develop a scheme for the next generation of drug repurposing. These drugs have the ability to restore homeostasis and to treat dysfunctional molecular pathways. This would be a huge step in the way towards producing drugs that can treat genetic disorders. The growth of human genetic resources provides the opportunity to better develop drugs. The topic of genetics is very important and can play a huge role in understanding diseases. If you understand genes and how they work, then you can better understand how diseases work and why and how. It is important that we have a great understanding of the topic of human genetics, but not even only to benefit us and our health today. Yes, it's good for us to know about the topic of human genetics in case we encounter some of these diseases or disorders in our personal life, in the lives of our family members, in the lives of our friends. But having an understanding of this topic can also be important for future endeavors. The topic of drug repurposing, which we covered in the last slide, that's something that they're working on and trying to change and trying to get changes on for future developments. So understanding genetics, understanding how genetics works, understanding how genes work will be very important in understanding the developments of the future. I hope this lesson was beneficial to you all. Thank you.